Good evening. For this project, we are doing a stop-motion project about the Meiosis model, and the materials we used for this project were just Google Documents, CapCut, and just the Windows Paint app. What is meiosis? Meiosis is a process in which a single cell divides twice to provide 4 cells. Unlike meiosis is a cell replication for gametes, otherwise known as sex cells, like egg and sperm. This replicates haploid cells to eventually produce a diploid cell. Before PMAP begins, we must start multiplying the DNA. DNA replication is the process where a DNA molecule makes an exact copy of itself, making 2 identical DNA molecules. The reason DNA replication is required for meiosis or any cell division is because it ensures each dotted cell produced during the process receives a complete set of genetic information, which allows for proper chromosal pairing. But now here, DNA replication begins in interphase, which is before mitosis. During interphase, the cell grows and DNA replication occurs. Cells also perform their normal functions in the phase. Once interphase is over, the cell transitions into the process of meiosis. The start of PMAT occurs, and the first main process is prophase 1. In this phase, the chromatin condenses into visible chromosomes by the homologous chromosomes pairing up, and the important process called crossing over occurs. This is important because it results in the genetic exchange between paired chromosomes. The next phase is metaphase 1, where all the replicated chromosomes line up in the exact center of the cell. This alignment is made by spindle fibers as they attach to the chromosome centromeres, as if it were a game of tug-of-war, to make sure chromosomes are disturbed properly. This is crucial as it verifies that everything is ready to be able to go into the next stage. In metaphase 1, there's also independent assortment, where the allele a cell receives for one gene does not impact the allele it receives for another gene, which occurs when the homologous chromosomes line up randomly causing a wide variety of possible genetic combinations. Anaphase 1 in meiosis is the stage where homologous chromosomes separate and move the opposite poles of the cell, marking the key event in the first division of meiosis, where the chromosome number is reduced by half, while the sister chromatins within each chromosome remain attached to each other. Essentially, it's a phase where homologous chromosome pairs are pulled apart by spindle fibers. After anaphase 1, telophase 1 starts, and in this final stage of the first meiotic division, it's where the separated homologous chromosomes reach opposite poles of the cell. The nuclear membrane reforms around them, and the cell prepares to divide into 2 daughter cells through cytokinesis, resulting in each daughter cell having half the number of chromosomes as the parent cell. Essentially, it marks the completion of the first meiotic division within the chromosome, now at the opposite poles of the cell, ready for the cell to split into 2 separate cells. After PMAT 1 in meiosis comes meiosis 2. This includes the previous stages in meiosis 1, that being prophase, metaphase, anaphase, telophase. But in meiosis 2, it is essentially the second division of the cell during meiosis, where the sister chromatids separate, resulting in 4 haploid daughter cells. Meiosis 2 begins with prophase 2. The chromosomes condense and a new set of spindle fibers form. Unlike prophase 1, homologous chromosomes pair up and undergo crossing over, and prophase 2 is individual chromosomes separate without any crossing over occurring. Now, metaphase 2 is where the individual chromosomes line up along the center of the cell, called the metaphase plate, where spindle fibers are attached to the centromeres of the chromosome in the centriole. Here in the metaphase 2, the chromosomes are ready to be separated into sister chromatids during the next phase, as the spindle fibers pull the chromosomes apart to the opposite poles of the cell. That is in anaphase 2. The next phase now is anaphase 2. In this phase, the sister chromatids of each chromosome are pulled apart and move towards opposite poles of the cell. The main difference between anaphase 1 and 2 is in anaphase 1, homologous chromosomes separate from each other, while in anaphase 2, sister chromatids within a chromosome separate. Okay, now, the last and final stage of meiosis is telophase 2, and in the phase, the chromosomes reach the poles of the cell, the nuclear membrane reforms around each set of chromosomes, and then the chromosomes then deconsent, resulting in the formation of 4 distinct haploid daughter cells, with half the number of chromosomes as the original parent cell. The main difference between telophase 1 and telophase 2 is that telophase 1, homologous chromosomes separate, resulting in 2 diploid daughter cells, while in telophase 2, sister chromatids separate, producing 4 haploid daughter cells with distinct genetic makeup.